The Story of Mipomersen. Despite remarkable advancements, heart disease remains a lurking danger that continues to pose a severe threat to public health. One such condition is homozygous familial hypercholesterolemia. To understand this cardiovascular killer, we must answer several questions and learn how mipomersen can help. To do this, we must step back and explore the world of lipoproteins. Our body utilizes various lipoproteins, each with its own function, structures, and composition. However, our focus lies on low-density lipoproteins. LDLs are cholesterol-carrying machines that use our blood vessels as highways to carry cholesterol to all areas of the body. A component of LDL is apolipoprotein B100, which allows LDL particles to deliver cholesterol by recognizing and binding to receptors throughout our body. LDL can also lead to atherosclerosis, a condition in which cholesterol accumulates on arterial walls and hinders blood flow to essential organs. Therefore, to regulate cholesterol levels, the liver expresses LDL receptors which can internalize and remove LDL particles. Familial hypercholesterolemia is a genetic condition caused by the inheritance of one or two mutated apolipoprotein genes, which disrupts the affinity of apolipoprotein B100 with the LDL receptor. This leads to extremely high LDL cholesterol levels and greatly increases the risk of serious cardiovascular disease.
The first drug designed for treating homozygous familial hypercholesterolemia was lomitepide. While lomitepide effectively lowered LDL levels, it was associated with severe side effects including liver toxicity, increased hepatic fat accumulation, and gastrointestinal disturbances. The discovery of mipomersen began in the early 2000s when INS Pharmaceuticals, in collaboration with Genzyme, embarked on developing a drug using antisense oligonucleotide technology. ASOs are short, synthetic, single-stranded sequences of nucleotides that attach to messenger RNA to alter or reduce protein expression. This approach in particular was adopted for mipomersen to target specific mRNA molecules in the liver to reduce apolipoprotein B production that is essential for the formation of LDL cholesterol.
Mipomersen is administered via subcutaneous injection, meaning under the skin. It's absorbed and predominantly distributed to the liver by binding to circulating plasma proteins. It's often prescribed as an adjunct to be used with other lipid-lowering therapies and dietary modifications. Mipomersen is an antisense oligonucleotide, or in other words, it's a short synthetic RNA sequence. It's specifically complementary to apolipoprotein B100 mRNA, selectively binding to it via base pairing in the cytoplasm of liver cells. The complementarity between mipomersen and ApoB100 mRNA results in selective mRNA degradation by the RNase H1 enzyme. Reducing ApoB100 mRNA reduces synthesis of the ApoB100 polypeptide. Consequently, this inhibition of protein translation results in a decrease of low-density lipoproteins and thus cholesterol in the bloodstream.
3 clinical trials showed promising reductions in LDL-C, ApoB, and atherosclerosis in mouse models of hyperlipidemia and LDL receptor deficiency. Therefore, it was given permission to be tested in humans. Phase 1's focus is on safe and effective doses. Studies found significant decreases in LDL-C and ApoB compared to the placebo group. However, some adverse effects were also observed. Examples included injection site reactions, flu-like symptoms, and elevations in transaminase liver enzymes, a sign of liver injury. Mipomersen was tested on patients with diverse medical histories. All studies showed a substantial decrease in LDL-C and ApoB levels, although liver enzyme increases 3 times higher than the upper limit were reported, and around 20% of patients discontinued the medication because of the adverse effects. In Phase 3, reduction of ApoB was found to correlate directly with an increase in liver enzymes. A rare side effect also presented, an increase in hepatic fat accumulation. Due to hepatotoxicity, the EMA never approved the use of the drug. In 2013, the FDA approved it as an HoFH adjunct therapy with a warning of hepatotoxicity. Currently, it is only available for special use through the REMS Drug Safety Program.
Mipomersen was discontinued due to adverse effects such as hepatic steatosis, where blocking ApoB synthesis impairs very low-density lipoprotein secretion and causes accumulation in the liver. One potential solution is concurrently treating this triglyceride accumulation by limiting acetyl-CoA carboxylase activity. Mipomersen also causes injection site reactions, which could potentially be mitigated by oral administration. However, this approach is likely impractical due to mipomersen's large molecular weight. Injection site reactions might also be mitigated by breaking up its weekly administration into more frequent doses, since biomarkers indicating systemic immune system activation were absent during trials. suggesting that these reactions are dose-dependent. The solution was also found to reduce the severity of flu-like symptoms. More research is needed to improve Mipomersen's safety, but it has great promise.